Okay, hello GCSE students and welcome to the Science Shack for this lesson on how DNA is structured and how DNA actually controls the cell. Now, since year 7, you've probably been told the nucleus of the cell is sort of the brain of the cell, it controls the cell, or if you've been a bit more sophisticated than that, you will have been told that it has genetic information, has DNA, store of DNA. Now, both of those things are true, but it isn't until this point in the GCSE, and we only really touch on it here, that we actually bring those two things together and explain how the containing of DNA can lead to the control of the cell. And I think this is a really, really good bit of the syllabus and starts to get into really exciting biology of genetics and an understanding of how we go together and how our evolutionary history works. So, we're going to crack on straight through these objectives. What is DNA? This polymer made of nucleotides, what is it? What the double helix is, the most famous of scientific images, the backbone, the bases, how they pair together, how DNA codes for a protein, and obviously then this idea, if we're going to put that in fancy terminology, in protein synthesis to a GCSE standard. Now, of course, I know most of you are going to immediately spot the gaps that I'm leaving out because this is GCSE standard. If you want to, I'm completely confident that most of you are fully able to go and look at the AS level version of this, which is basically the same process with a few of the blanks filled in. I know those blanks can be annoying when we teach this at a lower level, right? Particularly for those of you who are a bit more into your biology and thinking about taking it on further. So do feel free to go and look at the A-level versions. They won't be much more difficult than this, but they will fill in a few gaps. Okay, so moving on then. So first things first, this is the syllabus. As you can see, it just says the same thing as we just said there. You need to know that the bases, this language of DNA is in 4 bases, A's, C's, T's and G's. You need to know that DNA is a polymer made of lots of nucleotides, that DNA is actually a sugar, right? It's made of a nucleotide that's very similar to glucose. 3 bases coding for an amino acid, right? So that's how DNA codes for protein. The idea that long strands of DNA consist of this alternating sugar and phosphate, and the idea that we've got this repetition making a polymer. Okay, now the higher tier stuff, which of course is where most of you watching this video will be aiming, is essentially a simple description of how proteins are made, how it is that the code on the DNA becomes the order of amino acids in a protein, and how that protein then goes on and does its job. Okay, and of course you must remember complementary base pairing, which bases pair together always regardless of species, C's to G's, A's to T's. Now note there it says students are not expected to know or understand the structure of the RNA molecules, mRNA, tRNA, or detailed structures of amino acids and proteins. So those are the gaps that I'm talking about that we'll leave out. Obviously do go and skip ahead to the A-level versions if you are annoyed by the gaps that are being left out. So moving on. This is probably the most famous scientific image in all of history, I would argue, right? The double helix. The news pull it out whenever they want to talk about anything scientific. There's a science story and there's a big picture of the structure of DNA there for some reason, even if the story has absolutely nothing to do with DNA. But it's actually only within living memory that we know this really vital, important structure. Okay, it's only within one human lifespan before we had no idea that this is how DNA worked. Okay, so if we zoom in a little bit, put this in context. Now you will have all seen cells under the microscope. You will have all seen a nucleus under the microscope. You may also have looked at chromosomes down a microscope. But if we zoom in a lot on those chromosomes, then you end up right down to these things called histone proteins, around which is wrapped DNA. Okay, now to give you a kind of scale, you have 3 times 10 to the 9 bases, 3 times 10 to the 9 of these little approximately base pairs in every single cell. And you've got, I think it's 3 times 10, or 10 times 10 to the 11 cells in your body. So that means if you multiply those numbers up, you have more of these little bases in your whole body than there are stars in the Milky Way squared. Right? Squared. That's a lot of bases, and these are the code that spell out you. The only thing that's different between you or a plant or a bacteria or an insect is the order of these bases in this molecule of DNA. The molecule itself, same structure regardless of species, but it's the order that makes you different from any other organism. Okay, so let's zoom in right to one of those repeating molecules. This is called a nucleotide, and it is the monomer, the single unit, from which the polymer, the long chain, is made. And you're expected to know that it contains a phosphate, a sugar, and one of these base things that actually in their order spells out the code. Okay, so phosphate, sugar, base, that's the kind of GCSE version. You just need to know some shapes, that structure there. If you want to look at this down here, this is actually a bit more advanced. We've actually got some of the molecular structures in there, and this molecule here, deoxyribose, is a sugar. It's ribose, which is sugar like glucose or fructose or sucrose, with an oxygen taken away. So that's why it's deoxyribose. You find them in the nucleus and they form weak acids in solution, hence deoxyribonucleic acid, DNA. Okay, right, moving on then. So this is our overall structure, right, of our DNA molecule. So the monomer is the nucleotide. So one of these here, right, is a monomer, that's a nucleotide. Okay, and then when you join them all together, you get the polymer that is a DNA strand. Okay, you've got a little bit more detail about how that bonding takes place over here. But this side here is absolutely adequate for GCSE. In the middle here, we've got a series of bonds called hydrogen bonds that hold the two strands together. And you can start to see some of that pairing up that these bases do in this picture. Okay, so here are your key points. Nucleotides are joined in long chains. The two chains are joined together by bonds between the bases in the centre. Each backbone is this repeating sugar molecule, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and the bases join to each other in the middle. And you are expected to know that if there's an A on one strand, there's a T on the other strand. If there's a C on one strand, there's a G on the other side. Okay, we call this complementary base pairing, and like anything else in biology, it's due to shape. Okay, so you've got the shape of the A is complementary to the shape of the T. The number of bonds they form is equal and the shape fits. Okay, so like everything in biology, like enzymes or anything else, the shape is what's important. Okay, so if we label up this bond here, look, these are hydrogen bonds in the middle, right? That's our sugar, that's our phosphate, and those are our bases. Okay, it's called complementary base pairing, and you are expected to know that. The way I remember this is A and T, right, spell at, and C and G are my initials. So the initials of the guy that taught it to you. Okay, and then the last thing we need to do to get to that famous image, once we've got these two strands of sugar with the hydrogen bonds in between, is give them a twist. Okay, and that leads to the structure of the double helix. Okay, and you can see it labelled there. The bases in the middle, A's and T's and C's and G's, with the sugar phosphate backbone winding round the outside. Okay, right, so that's all great. That's the structure. That's a very quick run over the GCSE level structure. If you want to know more about the detail of those molecules, look at the A-level videos, right, because they will give you a bit more detail about some of the things I've just skimmed over there. Right, the order of the triplets in the order of these bases, right, on these things in threes codes for the sequence of amino acids. We call this the triplet code. Okay, so this here shows you as we go down the DNA, right, this GAG here, right, that triplet is referred to as a codon, and that tells a bit of the cell, right, that in that particular protein that this gene is coding for, there should be this amino acid, GLU, glue, right, CCC, PRO, that one's proline, this one's serine, isoleucine, so on and so forth. And if you think about it, you know which bit of the cell I'm talking about that reads this information and turns it into protein. Because since year 9, you've been taught about the ribosome, this bit of the cell that gets introduced at the beginning of the GCSE, and you get told this manufactures protein. What that's actually doing, and hopefully this puts a few things together, is it's taking a copy of the DNA's code from the nucleus, reading it, and stitching amino acids together to make a protein. Okay, and that is basically how this triplet code works. Okay, now each gene contains the sequence of bases that codes for one protein. In fact, that's what a gene is. When we talk about genes, we say a gene is a sequence of DNA that codes for a particular protein, and it's the proteins that make you you. Right? You've all got proteins for hair color, eye color, skin color, blood type, the enzymes that digest your food, the antibodies that fight off viruses. That's all in the news at the moment. This code is the only thing that spells out proteins, and it's the proteins that tell the cells what to do. Okay, so moving on then, right, into this one. As we said here, look, how does DNA code for a protein? Well, it's a triplet code. It's always 3 bases. Okay, it's like a language that's got words in it, and all those words are only ever 3 letters long. So a sequence of 3 bases of triplet code codes for a particular amino acid. The order of that triplet ensures the order of the amino acids are added in the correct sequence in that protein, which means that protein, has its particular job. Maybe it's an enzyme with a particular active site. Okay, so how does this work? Well, just to really hammer this home, 3 DNA bases spells out 1 amino acid. Okay, and that is called a triplet, a codon, right? And it's called the genetic code. And it is the ribosome that reads this molecule RNA and drags in the right amino acid. Now, this is one of those gaps I was talking about. You're not expected to know what RNA is. It is perfectly adequate at GCSE to say that the DNA's code tells the ribosome which amino acids to put in order. But those of you who are thinking, hang on a minute, isn't the DNA in the nucleus and the ribosome somewhere out in the cytoplasm? How does that work? You're quite right. There is an intermediate molecule called RNA. You don't need to mention that at GCSE. If you want to know more about RNA, then have a look at the A-level content. Okay, it's not too far away. So this here, right, I cannot really explain to you how important a breakthrough this code is. This, to a biologist, is basically our periodic table. This is the full genetic code, and it tells you which bases code for which amino acid. With this code here, you can genetically engineer anything you want, in theory. You can decipher any protein and work out what DNA coded for it. You can work out what any stretch of DNA would code for, in theory, and you can edit, you can tweak, and that is genetic engineering. Okay, now obviously some of you might notice this has got U's in it rather than T's. I've told you it's A, T, C and G, and this has got U's in it. That's to do with the fact that this is actually the RNA bit, that messenger molecule, which again I'm not going to linger on. So if you want to know why it's U's and not T's, go look at some more advanced videos. Okay, so here's the synopsis from a GCSE perspective of how protein synthesis works. This is a higher tier GCSE synopsis of how protein synthesis works. Right, so the genes make a template molecule, and saying a template molecule is fine for GCSE. That template is RNA, it's what it is, but you don't have to say that or know that for GCSE. Okay, so genes in DNA make a template. That then leaves the nucleus to bind to a ribosome. Right, you can see that happening down here. So the template's coming out of the nucleus, binding to the ribosome. The carrier molecule brings the correct amino acid in. Again, you are not expected to know about these carrier molecules at GCSE, just that the ribosome is able to bring the right amino acid in and bind it on to the correct triplet, like that triplet code. What then happens is that protein then folds up. Okay, the amino acids interact and folds up into a 3D structure. And that 3D structure tells the protein what to do. Think an antibody. An antibody has to have a very specific structure to bind onto an antigen. An enzyme has to have a specific active site to bind onto its substrate. And that is all comes from the order of the amino acids. Okay? And what happens then, of course, your protein has been synthesized and it goes off and it does its job. Which proteins you are producing depends on what type of cell and what type of organism you are actually looking at. Okay? So that's a very quick run through, right, on covering the idea that DNA is a polymer made of nucleotides. We've got bases A, C, G and T. They bond together, C to G and A to T. The idea that 3 bases code for a particular amino acid, and that basically is the basis of protein synthesis, and explaining simply how the structure of DNA affects the protein that's made. Okay, so if you want a bit of extension on this, because science is ultimately a human pursuit, I said this is within one lifetime, okay, that we actually found all this information out. These are the people responsible for discovering DNA, the structure of DNA, that famous double helix. And it all ultimately came down to this picture. Now these guys here, Watson and Crick, as kind of the standard household names of who discovered DNA. Okay, if you ask from a pub quiz, who discovered DNA? It's basically Watson and Crick. However, these guys, right, particularly her, Rosalind Franklin, made a massive contribution and generated this evidence. And there's a bit of a scandal and a sort of potentially story of 1950s sexism, misogyny in the scientific world, particularly between these two, that meant that her contributions were initially heavily marginalised. So if you wanted to, one of the most famous stories of scientific intrigue, data thievery, misogyny, sexism and feminism, have a look into the story of the discovery of the structure of DNA if you fancy a bit of an extension. Okay, thanks very much. That's all I've got for you today, and I'll see you again soon. Bye-bye.